Hi guys, so good morning. So last time we've learned about the cell and the cell structure and also all about the introduction of your biochemistry. But for today, for this session, we're going to talk about the transport mechanism. So this transport mechanism is actually essential when it comes to studying about drugs and of course into knowing where does your food go? How do you digest your food? uh, is it going to your stomach? And then after going to the stomach, where does it go? It goes to your small intestine. It will be liquefied, and then they will be formed into those monomers. So it will become glucose, it will become amino acids, or it will become glycerol and fatty acids. Now, these preparations or these monomers are the ones being absorbed by your cells. So usually your cells do have the receptors as we've discussed earlier, and of course it has this certain uh, look at it in our okay. So in here, your cells do have what we call the cell membrane, and usually they are your phospholipids bilayer. And this phospholipids bilayer, they are actually semi-permeable as you can see it. So there are spaces in between, and usually this thing or this model of your phospholipid bilayer or your cell membrane is called your fluid mosaic model. So it has been embedded as if they are mosaic together. I think you've done arts like mosaic before. And then they are floating in fluid. So they are called, this is called your fluid mosaic model. Okay? So this is essential to know because those spaces in between this circle, as you can see here, okay, there are spaces in between, minimal spaces in between. These are the ones allowing the entry of your glucose, your potassium, your sodiums, and other drugs or other chemicals that is essential or that are essential for the cell. Okay, so let's move on. So there are actually types or two types of your mechanism transport. So we have your passive and we have your active. So the thing that you should always take note when it comes to passive is that we are not requiring energy whenever we are using this one. And for your active process, of course, it is in your PowerPoint slide, we are always using the energy, of course, your ATP. So your ATP stands for, okay, you answer that. So that would be your adenosine triphosphate. Okay, so the ones that are in abbreviation, I think you should do your research sometime so that you will learn it. Okay, and that's it. We have two types. We have passive and active, and we will discuss those under these two processes. Okay, so take note. Usually, your passive process is the one or the concentration gradient of your chemicals are going downhill. So you have to imagine a ball. So let's uh, write. Okay. So in your passive transport, you have to imagine that you have a ball. So here, I am going to draw a ball. My God, I can't really draw. So going downhill. Okay? Because usually in your passive transport, the chemicals are being transported from a higher concentration going to lower concentration. And panumdumon nyo na lang guys, diin ang maskapoy, ang gasaka sa dalan, o ang gasaka sa hagdanan, or ang gapanaog sa hagdanan. So usually, in your passive transport, we are not using energy. Therefore, you are going downhill or you are climbing down the stairs or you are going down the stairs. So you don't require more energy or any energy with that kind of activity. Do you get it? Okay, so let's go with your active. While your passive is actually downhill, your active is the one going uphill. So if you have a ball right here, you must need to exert a lot of energy in order for this ball to go uphill. Okay? So that's the thing that you should really remember. So passive, the concentration gradient or your concentration or your solute moves from a higher concentration going to lower concentration without needing any energy or just minimal energy. And in your active, you are going from the lowest part of the hill going upwards. So therefore, you will need more energy. So That's how you will remember your passive and your active transport. Okay, so under that you have other things. So let's move on. Okay, so actually the one under passive, or the theory that we are using under your passive transport, is what we call your diffusion. And I believe that you've tackled this already in your science classes. So in your diffusion, so as I've said earlier, your passive transport is the one responsible into having this movement of your solute from higher to lower concentration. Okay, so as you can see here, okay, the fusion is movement of molecules down their concentration gradients from high to low. Therefore, you don't need more energy, and let's just have an illustration for you to really uh, analyze it, or, of course, visualize it. So, it's like this, the pellet in here. Okay? You have to imagine you have a dyed pellet, or butang ta, may ara ka lang something patulo nga coloring sa imo nga tubig. So, if you do it at home, you can use your McCormick coloring or whatsoever. So, just drop a little. So, that McCormick solution of color is actually the concentrated one. Of course, if you put it on water, it will eventually distribute evenly to all of your solution. So, from the concentrated one, when you put it into simply a diffusive medium, of course, it will just spread. So from a concentrated solution, you will become more distributed. You will become low in concentration. The color will be quite labnaw when we talk about it. So this diffusion is actually the theory that we are using whenever we are talking about, okay, passive transport. So let's move on. So usually, the homeostatic imbalance, or the clinical application of this diffusion passive transport in our life, is of course this one, as you can see in the example. So what if nasunugan ka balay and you are one of the victims, or you've seen some of the people getting burnt? So usually it is really dangerous when you are a burnt big victim, because the skin is our protective gear. So when we don't have our skin, we lose almost all of our fluids. And then what happens to our cells? Okay, our cells just it could crenate, it could lyase, it could absorb more water, so we won't know what will happen. Okay, so basically we need to maintain a certain tonicity or a certain tone for our cell in order for us to function well. So this passive transport, sila ni siya ang nag-regulate sa tanan-tanan tang mga processes. So, let's just move on for a while, and then we'll go back to that. So, we'll really explain it further. Okay? So, now, let's go to the different types of your passive transport. So, we actually have 1, 2, 3, and 4. So, we just have the simple diffusion. So, we've tackled already the theory of your diffusion. It's just the movement of your solute from higher concentration going to lower concentration. Okay, so now we have your simple, your facilitated. It's same with your diffusion. It's just that we have carrier mediated and channel mediated diffusion. It means that we are using proteins and other channels present in your receptor in order for us to transport a certain chemical or solute. Okay, in osmosis, we are actually talking about not the solute but the solvent molecules. Again, for an overview, in osmosis, we are talking about solvent molecules. Okay, the movement of your solvent molecules from a low concentration, okay, low concentration solvent, okay, solution going to higher concentration solvent concentration. Okay, so next we have what we call your filtration. So in your filtration, we are just using a certain force that is applied by someone, or there is a certain barrier that is in standard. So kumbaga gusto ko yabe ang size nga makasulod di 3 millimeters lang nga kind of solute. So the solute that will enter would only be 3 millimeter solute because of the facilitation of something that would be a barrier. So it can either be semi-permeable, permeable, or whatsoever, as long as it is standardized. Okay, So let's move on. I will just explain later about the osmosis and the other things here. Okay, so for your simple diffusion, it is just the same as we've talked about earlier. So it was just the movement of your solute molecules from higher to lower concentration. Okay? So usually, ang nagasulod sa aton simple diffusion or sa aton nga cell, you have to imagine, would be your non-polar lipid soluble. When we talk about non-polar, what does it mean? Okay? When we talk about non-polar, guys, we are talking about a a chemical or solute that is not charged. It is without charge, and usually they are lipid soluble. It means that when you put it or when you dissolve it into oil or any kind of lipid, it will just dissolve. So bear in mind that your phospholipid bilayer, the outer area of that and the inner area of that would be lipid. So basically, the things that are lipid soluble would be your oxygen, carbon dioxide, and your fat soluble vitamins. So they are the ones who are able to cross your cell membrane without any kind of special transport, just by having diffusion. Okay, so we'll continue, and let's put it here. Okay. So the example of that again of your vitamins would be your ADEK. Okay guys, ADEK. As in A-D-E-K. Loloy akong age. I can't really control the pad that I'm using. Okay. Okay. ADEK. Hindi ADEK ha, hindi tayo magpatokhang. Okay. So let's go back. So after that, let's go to the lecture. Okay. So that would be your simple diffusion. So let's move on to your facilitated diffusion. So in your facilitated diffusion, the one that we are talking about here would be usually a carrier mediated or a channel mediated uh, kind of diffusion. So what's the difference between simple diffusion? So in your simple diffusion, you are just basically bypassing the phospholipid bilayer without entering any kind of receptor, channel, or any kind of protein that you can find in an embedded kind of phospholipid bilayer. In here, as you can see in the example, this is the carrier-mediated facilitated diffusion into which you can only allow a specific chemical as long as it is compatible with your protein. Okay? So, hindi da kasulod ang potassium mo or sodium if it is not meant to be. Hindi mo na pagpilito ng hindi meant to be. Okay? So, wala is okay. So, that's it. Carrier mediated or channel mediated. When we talk about channel, it is like a canal into which it is also specific to the size of the solute that would enter. Okay? So, kung amo lang gina siyang kaigo da, amo na aton pasalod. Okay? It's nothing green. Okay. Next. So, in your osmosis, the one that we are talking about would be the movement of your water. It's all about solvent. Guys, do always remember that we have two parts of your solution. It's always in your basic science, your solute and your solvent. Solute is the one being dissolved into the solution and your solvent is the one dissolving the solute. Okay, that would be easy. So usually, si water lang man ang ginatunaw ta permi. Magtimpla ka juice, ang juice nga powder imo solute, water would be your solvent. So in osmosis, what happens is that we are actually after for the movement of your water. So from a higher concentration, water, diba, nga solution, it will go to a lower water concentration solution. So, sa utok mo na na, higher concentration of your solvent molecules going to lower concentration of your solvent molecules. Suli lang sa diffusion. Si diffusion, it's all about solute. Okay? So, from higher solute concentration solution going to lower solute concentration. Si osmosis ya solvent. High solvent concentration going to low solvent concentration. And usually guys, mabalaan nyo ni si osmosis nga, kung ginauhaw ka, amon siyang gina-apply ka sa aton lawas. If you are feeling thirsty, usually you are running out of water. it means that all of your cell are having increased solute inside because there's no water to dilute everything that is inside you. That's why you feel thirsty. So that's the time that you will take water from a petsil or anywhere nga makakuha ka tubig in order for you to dilute all the solutes present, specifically your salt inside your body. Okay? So next. So this is just an example. So as you can see in the screen, these are your water molecules going through your aquaporine channels. So usually your water goes in through your aquaporine channels and some just bypass your lipid bilayer. Okay? And in your osmosis, the difference would be again the solvent and also the presence of your semi-permeable membrane. And paano ito siya ma-apply sa aton kabuhi? So, ari guys, a living cell has limits to how much water can enter it. And water can also leave cell, causing cell to shrink. So, basic. If you're going to be dehydrated, your cells will shrink. And when your cells shrink, what happens? You can die, actually. Your cells will just be crinkled. Okay? Ara bala guys, nga ara ka lang gani sa swimming pool. Ara bala naghulom-hulom ka lang for pila ka oras. What happens? Okay. Your skin becomes crinkled or wrinkled or whatever they call it. So that happens when you are becoming too dehydrated. So some of your water is coming out of your body. Okay, you are donating some of your water out of the body. So that would be a trigger of dehydration. Amo na nga pag alsa mo sa pool, uhaw ka. Same with having diarrhea, having to vomit all the time. So all your water is being excreted. So it means that your salt value or your electrolytes value inside your body would be increased or other ang mga sugar mo taas. Okay? So you need water to neutralize everything. And a certain amount lang. Kung sobra man, hindi man pwede. So in here, change in cell volume can disrupt cell function, especially in neurons. Guys, kinahanglan ta paghalungan ng aton utok. Our cells in our brain are really susceptible to water. So, have you heard of what we call your hydrocephalus? So, hindi ni siya necessary nga kung hydrocephalic ka, guys, tsura mo dapat ang bata na lang dako-dako ulo. It's not like that. So, there are people who have undergone accident into which hydrocephalic sila. Kinagtalang ang tubig to, nagkadtuto, and we also use mannitol as a drug to relieve that pressure. If you watch Grey's Anatomy or any kind of Dr. Romantic drama, mention pa si mannitol. So that's how important the balance of your water and your salt in the body. And guys, usually we are talking about tonicity when we talk about osmosis. So as you can see, the definition of your tonicity is the ability of a solution to change the shape or tone of cells by altering the cell's internal water volume. And what could really be the most important part of your tonicity is the level of your sodium chloride. So, sino di kabalo sang normal? Ay, wala ko digaliga live sa inyo guys. So, who knows about the normal amount of your salt concentration in the body? For you to be considered isotonic, it means neutral lang imulawas. Hindi ka mag-shrink, hindi ka mag-bloated. So, how much salt can the body handle? Okay? Okay, since nga nag-search ka na da or whatsoever, the answer would be your 0.9%. It means that you will have only 0.9 grams of salt in every 100 ml of your water in the body. Okay? So, magsubra da or magkulang, you can either become dehydrated or overhydrated, leading to hydrocephalus or any kind of water retention moments. Okay? So, mangkot lang ka mo outside the guys of our lecture if you don't understand anything. Okay? So, kay basit-dasig ko or anything. So, next. This isotonic, hypertonic, and hypotonic solution are always discussed in your basic sciences. So, when we talk about isotonic solution, the concentration of your salt and your water is just equal. Or we can talk about, ringon ta na lang. Okay, in here. Let me erase everything. Okay. So, in your isotonic solution, iso. So, usually, ang kakatabo di is that, ano hambal to gani? Equal ang imo nga osmotic pressure. It means that you have equal concentration of your salt ang dako-dako ni water. Water in the outside area of your cell and in the inside of your cell. So you can do a basic experiment at home for your laboratory for this. And I think you've had this already in your anatomy. So again, your isotonic solution is all about having equal amount of solvent molecules to that of your outside and inside of your cell. So ari, water molecule siyang dagko. May duwa di ka water molecule, may duwa di, may isa di ka salt molecule. So it means that they are applying same amount of pressure. So dapat sa lawas ta equal halos tanan because it will cause danger sa aton lawas. Okay, let's go back to our lesson. So in here, amon siyang tsura sa aton red blood cells if ever, okay, nga na-dehydrate ka. So, kung tamaan gid kadamo sang asin na sa imo lawas, you will become hypertonic. Imo nga cell ga shrink. And I think who eats kimchi here or whatsoever, I cook kimchi always. So, guys, amo ni siyang theory. 'Di ba ang imong petchay dako-dako na siya usually nga dahon, and it will overflow in your labador and whatsoever. But when you put more salt, what happens? The lettuce or the petchay collapses or it shrinks, right? So, amo man na siyang gakatabo sa aton lawas. Kung tamaan na kadamo sa aton asin, all of our cells will shrink, will become dehydrated, kisa no rescue pa. May mga friends ko nga ila mama napatay due to dehydration lang, guys. So, it is really important to know the balance of your water and sodium. And guys, always remember that sodium goes really together with water. That's why kung ang nanay ta kag tatay, mga lula mo kag lula da, hypertensive, you should always advise not to take salty foods. Because kung damo ka asin, damo man tubig, damo man ang unod sang imo nga bloodstream, taas man ang imo pressure. Okay? So, that's how we relate it. Okay? And in hypotonic solution, it means that you have little salt inside of your body. So, since little lang imo salt, what would happen is that cells take on water by osmosis until they become bloated. So, ang himuon naman niya ni sang aton cell, suyupon niya naman tanan niya tubig. Because, mahambal siya nga, ay, hala, ang body, jutay na ng iya salt. So, we need to get some of the water from the body, tapos kita mabaton sang tanan ng tubig. Kay para, mag-normalize to sagwa sa bloodstream sang tao. Okay? So, in your hypertonic solution, you have to imagine this. Ari. Kaya para, di nyo na lang pag If you're talking about hypertonic solution, this is your barrier. This is the outside again of your cell. This is your bloodstream, for example. And this is the inside of your RBC. Okay. What happens in here? So, in hypertonic solution, usually, sa outside sang imo cell, tamaan gid kadamo sang imo nga asin. As in. Or i-letter X na lang asin. Para hapo sa inyo kabuhi. Okay. Damo asin. Hara. Damo asin. Sa sobra kadamo sang imo asin or imo sodium, may ara ka lang 1, 2. 2 solvent molecules only present in here, outside. So, inside of your cell, there is just about little nga asin. Tapos, damo-damo naman tubig. So, kung damo-damo ka tubig usually, guys, anong gakatabo? Imo tendency usually is to dilute something. Sakto? So, this is your RBC. RBC. Okay. So, ang gusto matabo ni RBC, hala, madonate ta tubig sa gwa. So that her blood or his blood will be stabilized. Okay? So, tanan ni mga water molecules, because osmosis, kinasturyahan tabay. So, it is about the movement of your water molecules. So, this is what happens in your hypertonic solution. A certain area is concentrated way too much. So, your water should be donated in order to neutralize it. You get it? So, next, let's move on. For hypotonic solution, I think what you should do is that, in your hypotonic solution, sulit naman niya. Sa hypotonic solution mo, you have, in the outside of your cell, you have so much water in your bloodstream. Ara. You have so much water in your bloodstream, but you only have little of your salt. You only have little of your salt. And then inside the cell, what happens is that there is increased concentration of your salt. Damo siya. 1, 2, 3, 4, 5. But lang guys, ako mag-gets yun eh. Kaya basta, mo ni siya ang akong drawing alaw-alaw ay. And then, there is it's still your RBC guys. Sorry, gudya, mabakal na lang ko isang do pen na kamahal pa. Okay. So as you can see here, there are only little water molecules. So what should we do? Of course, ang sa aton bloodstream na naman, suli na naman. It will donate all of its water, the ones in circle again, to transfer to your RBC, thus causing it to be more big or more bigger. Let's go back to the illustration here. Okay, you can see now. So in your hypertonic solution again, your RBCs have little salt little salt concentration, thus ginadonate itong iyang water outside your bloodstream, causing for your cell to shrink. In your hypotonic solution, as you can see, is that you have more salt concentration inside your RBC and little of your water, thus your bloodstream donates more of its water going to the cell. That's why it becomes bigger. So that is the application of our tonicity in the body. So amo na guys, mag-dextrose sa inyo, basa-basahon nyo man. It's actually 0.9% sodium chloride. Okay? That's it. So let's move on. So for filtration, the only thing that is unique about filtration is that it's still diffusion, but we are using the pressure of your hydrostatic. So syempre, it's water. And it's only specific onto what size of your particle could enter. Kung gusto ko 3 mm lang enter, 3 mm lang because I only have this kind of filter. Okay? That's it. So, infiltration, usually we are applying this in our body through the kidneys, specifically in your Bowman's capsule. So, as you can see in here in the illustration, this is our bloodstream and this is the Bowman's capsule. So, our bloodstream is being filtered via this Bowman's capsule. So, usually, si Bowman's capsule ta, gusto yung mga gagmay lang gid ang masulod. So, it allows only your sodium, your creatinine, your little small sugars, and other things nga pwede maupod sa urine. So usually ang mga amino acid mo guys, wala mo na ginapangihi. Kaya kung may amino acid na kag kung may mga bacteria, usually it means that something is wrong with your kidney. Okay? So filtration, because it only allows a certain size of a chemical to pass through. Okay? Next. So I think the second one would be your active membrane transport. So in your active membrane transport, we have two. It's actually the main active transport. You have your primary and secondary under that here, and your vesicular transport. So why is it, or how is it different from your passive transport? Again, it's all about ATP. And it's all about electrochemical gradient, and it's all about the presence of your carrier-mediated proteins and your channel proteins. And when you ask me, Timis, sa passive may amuman to. So the thing is, we are talking about the sizes of your solute. So if your solute is too large for a channel, usually we use your ATPs. Or we use your active transport in order for them to go inside. Usually, mga antibiotic, guys, tag-as ni siya, dagko ni siya ang ilang molecular weight. So, solute is not lipid soluble. So, syempre, hindi siya kasulod because ang aton phospholipid bilayer is a lipid. So, therefore, butang mo do, nagbutang ka lang tubig sa imo mantika. It will not be compatible. Okay? And solute is not able to move down concentration gradient. So, we will explain this further later. So, in here, you have your active transport, kadamo balasahon, but we have two types of carrier proteins nga nagabulig sa aton sa active transport. We have your anti-porter and you have your sym-porter. When we talk about anti-porter, it's that you are carrying two different chemicals in opposite direction. And in your symporter, you are also bringing in two different chemicals or same chemicals in the same direction. So let's just look at an illustration here. Okay, it is an antiport and your symport. So in antiport, the other one goes in and the other one goes out. And usually an example of your antiport would be your sodium potassium ATPase. Again, the example would be your sodium potassium ATPase. So, kasi hindi na makamula it's sodium. Sorry, gidyo guys, ang aking potassium ATPase. Let's just bear with this kind of, if you can suggest a good whiteboard into which I can really write fast or whatsoever, I can suggest. Okay, sodium potassium ATPase, that's how you spell it. Okay, so let's go back. So now in your symport, it's just that you are importing your sodium and glucose inside of your cell. Okay, so you are going through a unified direction. So one direction. Shut up. Okay, so in your active transport, you actually have your primary and secondary transport. So usually guys, ari lang ang difference. In your primary active transport, we are dependent on your ATP. Again, we are dependent on your ATP. But in your secondary active transport, we also use a certain energy but not specifically your ATP. What we are after in your secondary active transport would be your electrochemical gradient. Imagine mo na lang, it's like the flow of your electricity. If one goes in, kung si sodium kumasulod, si glucose kumasulod, may electricity na itong nagaiban-iban, kag may mga potential charges sa imugwa sang cell, nga nagaproduce energy in order for you to have that cycle, the domino effect. When one goes out, the other goes in. So, the cycle repeats until nga may ma-generate kada nga magnetic force, okay, or electrochemical force. So again, primary active transport, ATP, secondary active transport, you are dependent on the flow of your ions. But it's not, sa aton nga illustration di guys, it's not necessary nga kung antiport ka, primary ka gid. If you are symport, secondary ka gid. So this antiport and symport are just examples of your carrier proteins. Okay? They are just example of carrier proteins that could facilitate your active transport. Okay? Are we clear? So in antiport, sodium potassium ATPase. In your symport, usually it is your sodium glucose symporter. Okay? Example lang sila isang carrier proteins. So here's your electrochemical gradient. So you just read upon it. But the thing is, kung may move, usually naga-generate ka electricity. So you are generating action potential whenever there is movement. Okay? And we've talked already about this. The sodium potassium pump is one of the most popular pump, okay, or one of the most popular protein area that could facilitate your active transport. And you will know in your pharmacology or in your anatomy that your sodium potassium pump plays a large role when it comes to our body function. So siya gining number 1 nga nagapa giyo sa aton nga ang aton nga cell ga generate electricity and all that. Okay? So here's an example. So the color blue actually is your potassium, and the one that is in red would be your sodium. So ara gabulus-bulos lang sila. When another goes in, the other goes out. So amo na siya daning kinanan yung mabalaan. Dito na siya pag-i-deeper because it's in your pharmacology. So next, let's go with your vesicular transport. So for your vesicular transport, amo na siyang third type actually guys ang inyo active transport. For your vesicular transport, actually, the one that we are using here would be a vesicle. So may ATP ka kag may vesicle. Magsiling ka vesicle do may salakyan ka. May ara may gasugat sa imo. Wow, social. Kinahanglan gid ni siya during MECQ. Okay. So there are two types of your vesicular transport. It is your endocytosis and your exocytosis. And I think you're really familiar with this. So endocytosis, it means that you are going into the cell. Exocytosis, it means that you are going outside the cell. Exit, exo, endo, inside. So when we talk about phagocytosis, it's all about cell eating. And when we talk about pinocytosis, it's all about cell drinking. Okay? So let's have an example. So let's not really read upon it. But in endocytosis, for example, cell mo ni siya. Amoy siya tanan. This is the whole cell. If this is the whole cell, for example, you have a bacteria going in, so the ones in red. So it will just enter the cell and then it will be surrounded by a certain protein coat. And then inside that, it will travel inside the cell and then it will come in contact with your lysosome. Remember lysosome? And then what will happen? It will digest that bacteria, or maybe it will lead into excretion of that bacteria into the cell. Because, ano, hindi ka diya belong sa sulod sang cell. Why is it that you're here? So, that's the thing that would happen during endocytosis. So again, in your endocytosis, it's either you go into the cell and then sugaton kasang protein coat, you will meet with the lysosome and then you will be digested. So it's either phago or pino, it's up to the solute nga gina-pa-enter. So if you are solid, usually ginakaon, phago. If you are in liquefied form or naka-water kada, be liquefied glucose kada, we are using pinocytosis. Okay? So just read almost. So, amo ni siya ang vesicle nga na-form during phagocytosis. It would be your phagosome. Okay? And here, these are just detailed things. And in your pinocytosis, it's just the same. It's just that ang imo nga gina-ingest would be in fluid phase. Okay? And lastly, we have your exocytosis. And usually exocytosis, we are using this one when we are excreting toxic substances inside our cell or in our body. And usually we are using this kung nag-exercise ta sa aton armpits. So usually we have discharges in that area, and exocytosis is the one that we are using. So we are actually using the secretory vesicles in order for us to secrete those toxic substances. So usually kung gaswet ka during exercise, that's good because all of your toxic substances inside the body would be carried out. Okay, so it's like cleansing. Okay, it's detoxification. So we are using exocytosis when it comes to excretion. So here's an example of your secretory vesicle. Usually it can be found in our armpits. So usually may mga discharges kita daya. That's why we use deodorants and other stuff in order for us to control odors and other secretions. Okay, so that would be it. So we will have a review with this topic. And ang active transport ta guys, when we summarize it again, it would just be the main active transport having your primary, secondary, and then your vesicular transport. In your primary, we are basically using the ATP. In your secondary, what we are using is actually the electrochemical gradient in order for us to produce an action potential. And the third one would be your vesicular, because we are literally using your vesicles or this carrier proteins or your protected coat nga nakita tato sa aton diagram. So under that vesicular would be your exo and endocytosis. Endocytosis, under that would be phago, your food eating, and your cell drinking, which is your pinocytosis. And your exocytosis is all about excretion of your toxic substances in the body, which usually happens in your armpit areas and other parts that have many discharges. So, pwede man sa ilong area. Okay? So, that would be it for the whole mechanism transport. So, if you don't understand and hindi ko dyan anything, wala ko naman intindihan. But if we have some confusing area that you need to clarify, just message me. Since this is just a hurried kind of tutorial. And me too. Maybe I'm kind of confused whenever I'm talking because, of course, I have many things to maneuver in here. So that would be it. I hope you will watch this lesson again and again and read your ink so that we will be in sync. Okay? So, thank you.